We are fortunate to have Alexander Ioannidis, or Alex, maybe you can tell us how you prefer to pronounce it. There are many different ways.
That's great.
Okay. Alex is a former postdoc in the Bustamante Lab with me, actually. And before that, he got his PhD at Stanford in computational and mathematical engineering. Now he is an adjunct professor in the School of Engineering at Stanford, and he teaches machine learning and data science. He also has worked in the past in superconducting computing logic and quantum computing at Northrop Grumman, and he graduated summa cum laude from Harvard in chemistry and physics, and had a master's in computational biology and a diploma in Greek from the University of Cambridge. So Alex has a diverse and very deep background in all of the topics that he's covering, and I think has sort of brought them together in I would say really a research program of ancestry methodologies and students that you've worked with. So it's been just a pleasure to get to know you and to get to know the work that you've been involved in, Alex, and I'm excited to hear about it today. So without further ado, please go ahead and present, and then how much time do you think we'll have for questions at the end?
How much do you usually want to have? About 10? 15?
Yeah, 10, 15. Yeah, that'd be good.
Yep. Okay.
So I know you have a lot of content. So, and we did send out the links to Alex's methods papers as well. So, so that you have that for reference. And this is being recorded.
Great, thank you. So I'll stop around 9:45 California time.
Perfect.
Precious.
Okay.
Great, thank you, Alice. I really appreciate your inviting me to present here. I think this is a topic is very timely as we're starting to see biobanks coming out with large numbers of whole genome sequences. It's becoming really important to develop ancestry tools that can accommodate that, because the existing tools cannot. For those of us who have tried working with many samples, once you're in the 10,000 sample range, and especially for whole genome, current methods really choke. So I'm going to talk about the methods development and the new methods we have that work really nicely for large-scale biobanks, and they're actually more accurate for whole genome than any of the existing methods anyway. So they're faster and they're more accurate. But I first wanted to talk briefly about why genetic ancestry, and maybe everyone who's on this call already knows that it's interesting, and that's why you're here. But I just wanted to mention. So here's a PCA of patients actually at Stanford Hospital, and these are patients who were COVID positive, and we had them as part of a pandemic tracking strategy. But I just wanted to show you that we have a very diverse cohort at Stanford, and it's genetic ancestry that kind of allows us to visualize that. And here you see that the different clusters, and there's a number of reasons you might be interested in genetic ancestry. It may be just visualizing the diversity of a cohort. It may be that you're interested in doing GWAS correction. It may be that you're interested in the problems of portability of PRS scores. And admixture mapping, which is another useful tool. And here's an example of admixture mapping on that COVID-19 data set. So this looks at severity score regressed across the ancestry along the genome. Sorry, I think. And what we see for each ancestry, we can see where having that particular ancestry in your genome, and the x-axis is looking along your genome, is associated with, or was associated in our cohort with, having more severe COVID infection. And you can see actually there's a locus here on chromosome 14 that having Oceanian ancestry was highly associated with having a more severe COVID reaction. So that's interesting, and we can map things that way. We can also look at clustering here. This is looking at ancestry clustering superimposed on the viral genome tree. So of course the COVID-19 virus is mutating as it's spreading through the community and forming clusters, and we can look at how it's spreading through the community if we can label the leaves of this viral tree with the genetic ancestry of the people who were infected. Now, there could be better ways of doing this. Certainly, if we had more data, it would be nice to know the locations where people are living, and it would be nice to know the their own self-reported ancestry. But in many cases, we don't have that. And the interesting thing is, just with a swab, we have all of this information. With a nasal swab, we've got the viral genome. We also have the host's genome, so we can get the genetic ancestry. And just with a nasal swab, we can now trace how things are spreading in this way. So a lot of interesting things about genetic ancestry. And what is it? So it's something that kind of emerged through the historical process of humans spreading across the globe and moving to different continents. And there's always been, of course, movement back and forth, and that's very interesting and something I like to look at. And admixture has continuously existed. There's never been such a thing as a pure population of one type or another, but still we have these separations, and with time, and that's what we're showing here, that each of the populations executes a random walk in frequency space. The frequency of their alleles changes somewhat independently as they tend to be less connected to each other with less migration in between. And that causes ancestry clusters to emerge. And then, we can look at this in terms of the standard equations of population genetics. I just want to throw this up there to say, okay, it's genetic drift that is governing most of this. It's not selection, it's just a random walk through frequency space. And it produces something like this. So this is a principal component analysis of a number of individuals. And we're looking at 3 principal components. Sorry, we can see the different ancestries are colored here, but you can see that these colored clusters, they don't really reflect the full reality. There's a continuum, right? So there's people lying all in between. But for convenience sake, it is nice to label some of these clusters to be able to talk about things. This is something that people are discussing better ways to do it, so as not to put labels on people, because as we can see, there aren't clear distinctions. But still, one of the very useful ways that people do this is to look at cluster centers and talk about fractions. And so you can see these dark dots are cluster centers that are found by a clustering algorithm. In this case, admixture is the algorithm. It's a very commonly used one. So you see the cluster centers superimposed in this PCA space. So you've got a continuous space, and then you have a way of trying to label points. And here the points are colored by the cluster center they're closest to, and as a way to make it easier to represent these high dimensional spaces. And so here is a representation of that plot, the same plot I just showed you, but shown a different way. So this is the full continuous plot. Instead, we can take every individual who's a point on this plot. We can represent them as a vertical bar, and you can see there's a lot of very thin vertical bars here. And if we look at all those vertical bars and we look at the coloring, then we are looking basically at a fraction for each individual of assignment to each of these colored cluster centers. And that fractional assignment lets us basically have a way of describing in a simple representation where people lie in terms of this space. So what fraction do they have assigned to each of these clusters? Okay, this clustering algorithm is called admixture. It was first brought out as a program called structure. And now we've introduced neural admixture, which lets you do this, but with hundreds of thousands of samples. So I'll be discussing that. But again, why would you want to do it? Well, here we have it again with that COVID data set from Stanford Hospital that I worked on. And we can see every individual in the data set here, and we can see their assignment, and we can see patterns emerging, right? It's a visualization and a descriptive strategy. And we can see our European patients, we can see our Hispanic patients. They're the ones who have quite a bit of this light blue, this sort of sky blue ancestry cluster assignment, and that's one that's associated with Indigenous Americans, and a lot of Latin American and Hispanic patients have that Indigenous American ancestry. We can see that there are a lot of patients coming with that background, and reflecting the fact that the COVID was spreading amongst populations that were least able to quarantine because they were having to still go to work at the time. So why else do we want to do ancestry? So I want to show you the other type of ancestry that's important, and it's called local ancestry. So local ancestry is where instead of looking at every individual as a single dot in a PCA space, right, which was what we were doing here, and then coloring them, we look at their genome and try to determine along their genome every segment which ancestry background it came from. And of course, as populations become more and more mobile and admixed, we get more and more individuals who come from multiple ancestries, and we get these sort of segments that in their genome that came from different backgrounds. And this is an individual from Easter Island on a paper that I worked on where we showed that in Easter Island, you actually find, besides Polynesian ancestry, Easter Island is a Polynesian island, and European due to colonial activity, we also see on this genome of this individual some green Native American ancestry. And this was a really interesting question. Why is there Native American ancestry on Easter Island? It turns out there's Native American ancestry on a few other Polynesian islands. And by looking at these segments along the genome, we can do all sorts of interesting analyses, and I'm showing you that here. This is simplex plot. We can look at the length of the different ancestries and their mixing. And basically, by looking at these distributions, we can figure out how many generations ago this ancestry entered, and we can do historical analyses. And it turned out here in this paper that actually the Native American ancestry entered in the Middle Ages. And so there was contact in the Middle Ages between Polynesians and Native Americans, which was an interesting thing. So you can get these interesting historical perspectives also. But what I want to emphasize here is that we have to look now at just the segments of Native American ancestry if we want to focus in on the genetics related to that background. And we can do that with local ancestry inference. We can find just the segments that are Native American, and then we can do analyses that are specific to that, and that's what you're seeing here. So here you're seeing across all these different Polynesian islands, looking just at their Native American segments and determining where that Native American ancestry came from within the Americas. And we can do that. We pinpointed actually to somewhere in the Colombia-Ecuador region. So that's where the contact between the Polynesians and the Native Americans occurred. That's interesting historically, but we can do a lot more with that if we're trying to do genetics. So we can also look at IBD, which is shared segments that are shared because they came from the same ancestor, and we can look at those in each of the different ancestries. So we can look here. We're looking at a network in green of the shared segments that are in the Native American ancestry. And it shows that, there was the shared pattern in these most remote Polynesian islands. They all shared ancestry from the same Native American contact event. So, some sort of trade network, or more likely a single population that contacted the Americas and then later settled all of these islands and took that ancestry with them. So with local ancestry inference, we can zoom in on just the specific question related to a specific ancestry by looking at just those segments. And we can start to look at just those segments in terms of their IBD, just those segments in terms of where they could have come from against a panel, let's say, of other samples in the Americas here, just those segments in terms of how severe the COVID infection is in individuals who have a segment at that position in the genome, and that's what I showed you here. So we do these ancestry-specific analyses, which can be quite powerful. So that's the reason local ancestry inference is interesting. Here's another example with populations more relevant to us here in the United States, which are Latin American populations, Hispanic populations. So looking just here at their East Asian ancestry, we were able to determine the sort of trade route that brought the Filipino ancestry actually to the Pacific coast of Mexico during the Manila Galleon, another historical episode. And then finally, we can look at very, very detailed effects, so founder effects, which are very important, of course, as we know, for populations, bottleneck populations. Ashkenazi Jewish populations are one of the ones that are best studied that have these strong founder effects that lead to strong genetic surfing and genetic drift. But we can look at these in Polynesia. This is another place, of course, where we have strong founder events. And that's what we did in a paper just came out last month that, again, had to focus on just the Polynesian segments in order to understand these founder effects, because now across these Pacific islands where we investigated this, there's a lot of European ancestry too, and the European ancestry doesn't show the same founder effects as the original foundation of the islands by Polynesian sailors 1,000 years ago. The European segments obviously are showing different colonial effects. So we have to do local ancestry and isolate just the Polynesian ancestry to start understanding these founder effects, which are of course very important for understanding genetic disease in these populations. And this is just a plot that shows the really strong climb in founder effects, getting more and more bottlenecked as you move out across this settlement pattern across these islands. Okay, so that's why local ancestry is interesting, and I showed you previously why global clustering is interesting, and now I'm going to talk to you a little bit about our algorithms to do this. Okay, so the first algorithm is for local ancestry inference, and this is designed for large-scale biobanks, but it works for any data set, and since it's more accurate than any existing methods and faster, I'd advise you using it. Of course, we developed it, so I'm going to be selling it, but I really think it's ideal for using in any case. So we're calling it Gnomix, and it's designed to be sort of a legacy to previously existing methods. There was a method that was very commonly used called admixed that used random forest, and we try to match our output in our input styles to that so people can plug it right into their pipeline. But it's a very different algorithm under the hood. And you can kind of see an outline of it here on the right. So we start with the DNA sequence, or the letters, and we translate them into a binary sequence, zeros and ones, which we can do by comparing to the reference, right? And so we have a reference all 0 versus 1. And then we apply various modalities I'm going to show you in a moment. So we should really see genomics as a Swiss Army knife. We have a base classifier, in this case illustrated as a tree, classification tree. So boosting is one of the better methods. And then on top of that, we have a smoothing classifier, and this is sort of novel. All the methods before had looked at actually a fixed model, either an HMM or a conditional random field, but we actually learn the smoother instead of having a fixed model. What that means is that, first of all, you do not need to specify how many generations ago you believe the admixture event took place, which generally you don't know, and may vary across your cohort in any case. It may be many different historical events and different people. And you actually can also, by learning this smoother, you can learn where different ancestries are more commonly to be found switching from one to the other. It may be that in this population, it's more common to see a combination, let's say, of Native American and Polynesian than Native American and African or something. And all of this can be learned. And then you get your final local ancestry inference. But each of these models, the base and the smoother, are things that you can stipulate in the program. So we've implemented a whole host of different machine learning methods. We have the recommended default for whole genome, a recommended default that's different for genotype array data. In that case, we recommend a novel string kernel method we developed. But if you have more SNPs, which you do with whole genome, then it's better actually to use a different technique, which is our whole genome default. And then we have different smoothers. Again, a recommended one for high-speed performance, for most accurate performance, but there's a whole host that you can use. So you could see this really as this genomics as maybe 20 different local ancestry inference algorithms in one, and you can choose which of those 20 you want. And they're all sort of optimized to be the best at whatever particular application you may have in mind. I wanted to call out the two students who did the work on this project. Arvind Kumar and Helgi Hilmerson, really great students in the department I'm in computational mathematical engineering, and they did some really nice work on this and learned a lot about genetics and population genetics in the process. So this is kind of the important figure if you're trying to figure out why you should use our technique. So what you see here in the top, I'll just walk through it slowly because there's several panels, right? But in the top, you've got the in red, various shades of red. You have the different machine learning models that exist in our new technique in genomics. And as I mentioned, it's a Swiss Army knife. You can pull out whichever of these you want, and you can see the performance of those versus the performance of ELAI, LOTER, and RFMix. Those gray dots are existing local ancestry inference methods. You don't see loader here because we couldn't even run it. It took so long, it was just impossible. And our methods are up here. So accuracy is on the y-axis, so higher on the y is better, you're more accurate. Speed is on the x-axis, so further to the right is better, is a faster model. And that's a log scale, by the way. So this is dramatically faster the further you are to the right. And you see so you really want to be in the upper right-hand corner, right? That's the most accurate and the fastest methods. And you can see all of the different techniques in our Swiss Army knife of techniques we have in genomics. Those are the red ones. They're all here at the top. They're the most accurate and the fastest, and quite a bit more accurate and quite a bit faster than the existing gray methods. Again, remember, this is log scale. So we're talking about something that, in those Polynesian papers that I showed you, this is true, took us 2 weeks to run on a data set that was in the hundreds of samples, would take now on this mere minutes to run. And if you wanted to run on tens of thousands or hundreds of thousands of samples, you could do it in a day for 100,000 samples. So this is a dramatic improvement. You could never have run 100,000 samples with these old techniques in gray. And they also choke on whole genome. They're really designed for genotyping. So that's great. Here in the bottom left, we're showing all the different models that we've implemented in this sort of Swiss Army knife technique, genomics. So we've got light GBM, we've got k-nearest neighbors, we've got naive Bayes, we've got the SVMs I mentioned. We customized one called the covariance random string kernel, which is really, you see, just squeezes out a little more accuracy. So that's the one we recommend and we describe in the paper, but we've implemented several standards SVMs. And then we have, the traditional random forest that was an RF mix. You can see that the accuracy is very low for that. And that's why we're, not using it, but it's there for comparison. LDA, we tried a whole bunch of things, but as you can see, there's only a few that really define the Pareto optimal frontier. And those are the ones that we recommend as default for different applications. And then the last performance plot is this one in the bottom right. And I think this is really impressive, actually. So this is showing you the performance of sort of the existing standard that people tend to use, which is called RF mix, versus Gnomix, our method. And what you can see in solid is performance on whole genome data, and dotted is performance on array data. Red is our method, the best version of our method, and gray is RF mix. And you see that our method does better, and accuracy is here on the Y, so being higher is better, does better on array data than RF mix does on even whole genome data. So even with all of the entire genome, all of these features, RF mix still does worse than we do with only a tiny subset of all those features. That is, just array data. But of course, if we have whole genome data, then we do even better. And you can see the gap is even larger. That's the solid red line here. And what we have on the x-axis is the number of generations ago that the admixture event occurred. So again, as I mentioned, these admixture events, typically they occur throughout history at different times, but we simulated individuals from real genomes of real individuals. So we just simulate the recombination process, so it's pretty well understood, right? We simulated individuals and their progeny over many, many generations to see how well we can identify segments that were the product of mixing that occurred many generations ago. And if something occurs many generations ago, then these segments from one ancestry or another get broken up into very small pieces by recombination over many generations. So you get very small segments after admixture events that occurred far in the past. And that becomes a more challenging problem because a small segment is harder to identify in the genome. And so you can see as the generations get larger, so that as we're looking further back into the past, you can use as a rule of thumb 30 years per generation for the average human, averaged across all the children that a human has historically, and both male and female parents, sort of a well-established number. So just take about 30. So if you multiply by 30, you see we're going back to 3,000 years ago. So once we're 100 generations ago, we're looking at events that happened 3,000 years ago. We're looking at very old segments in the genome. And you can see that genomics has still got 90% accuracy, even 3,000 years ago, which is incredible. You could not have used local ancestry in the past for events that were that old. And maybe you're not interested in events that are that old. That's fine. Most admixture events, colonial admixture events that happen happened in the last 500 years. Things to do with the transatlantic slave trade, things to do with indigenous Americans interacting with European colonists and things like that. Well, in that case, we're looking at things that are more like, maybe 20 generations ago at most, and you can see that genomics has got extremely high accuracy at that point. But accuracy is not the only thing. The other thing is speed, and as I mentioned, for speed, it's not even possible to run any of these other algorithms when you have hundreds of thousands of samples. Now, there's one last point that I want to make, which is why our methods are colored different shades of red and the existing methods are in gray. So the red here bar you see on the right is colored with darker intensity depending on how large the model size is. And what does that mean? Well, these existing techniques in gray do not let you save your model. So that means that every time you need to rerun your local ancestry inference, you need to retrain it. So machine learning algorithms need to be trained. That takes a lot of time. It also means that you have to have on your computer, your server, all of the reference data that you're using to train the model. So you have to have, a number of samples that are European, a number of samples that are, let's say, Native American, a number of samples that are African, in order to train your model to recognize those different ancestry segments. And those individuals have to be completely of that ancestry. They can't not be mixed, because then they will not work for training samples. So it can be kind of hard to locate enough training samples in many cases, and it requires some knowledge of population genetics. And besides the fact that this is computationally expensive to retrain every time, it also means you have to have someone trained in your lab who knows how to find these samples and acquire them. And it may be that they're even difficult to get access to data sets is often not trivial, as we all know. Well, the nice thing about genomics is that once you train a model, you can save the model and you can send it to somebody else, and they can run this analysis without having any of this reference data, without having to do the training, any of that. And in fact, we provide on our GitHub, and I encourage you to go and explore it afterwards. The GitHub is AI-sandbox on GitHub, AI-sandbox. We have Gnomix there and neural admixture, which I'll talk about in a minute. And we have our models there. You can download a whole genome model right now and run this local ancestry inference on your whole genome data to identify Native American, African, East Asian, European, Oceanian ancestries. It's already trained for you. You don't have to do any of the obtaining of data sets for references or thinking about how to merge them or any of that. Just download the model and run it. And that's what we're showing here. And the darker models, the darker red, just are larger. So it's nicer if the model isn't so large, right? Because then it's easier to store it on your hard drive and download it. And so we're showing that some of our methods have lighter, smaller models than others. But the portability of the model is really an important thing, especially when data has restrictions on being shared. It's really nice if you can just train a model and then rerun that, and you don't need to share the data. All right. So so then we'll just quickly look at how this plays out in practice, the mechanism under the hood. So for genomics, we have this base classifier first, this machine learning model that goes along the genome and identifies for every piece of the genome which ancestry it believes that piece came from. And that's what you see colored in here. The ancestry color codes are here. So that's for a person, a piece of a genome. And then the smoother comes along, and it's a different machine learning method that learned how to identify transitions and properly classify and smooth things out. And that, you see, does a lot of error correcting. And so there's a lot of these small slivers of misidentified ancestry that the first method makes errors. And then the smoother comes along and corrects almost all of those. And you see the only error left, here's the truth, is the bottom colored in segment of the genome. The only error left is this one misidentified European segment. So it's very, very accurate. And we're showing here the probabilities of the model and how they vary, and the confidence and all of that. And this is sort of interesting. The lower plot, we show the smoother model, how many neighboring pieces of genome it needs to look at to try to do its smoothing. And it turns out, depending on the ancestry, it may need to look at a wider part of the genome, or it may not. And so for some ancestries, it can classify very accurately by looking at just a very narrow part of the genome. And those are for ancestries that it's very, it has a very easy time classifying them. And you can see that here for African is a very narrow area that the x-axis is how far along the genome the smoother needs to look. It's a very narrow area that it needs to look at, and that's the dark shaded area, in order to properly classify. And you can see for other ancestries, and for European is a good example, it needs to look at a much broader area. And the reason it needs to do that is because it's confusing European with a different ancestry. And so it has to look at a lot of the genome in order to make sure it doesn't make a confusion. And I'll show you in a minute what ancestries it's confusing it with, but it's actually this one. We put in an ancestry we called West Asian, which is basically Middle Eastern, and as you might imagine, that is very close to European, especially Mediterranean European individuals, and so that's a very subtle distinction to make, and so more of the genome has to be scanned in order to get that accurate. But we are able to get that at very high accuracy, above 95%, and that just shows how strong this method is. It can separate even sort of more closely related populations. Okay, so this is another point I wanted to make. So the existing local ancestry inference methods do not do this. So this is called rephasing. So as you may know, if you run a local ancestry inference method, you have to first separate the two paternal chromosomes, right? Because they differ in ancestry. So you have to separate for an individual which of their SNPs are coming from their mother and which are coming from their father. And that's called phasing. There are statistical phasing algorithms out there. They work decently for nearby SNPs using LD and correlation, but for long-range segments of the genome, usually at some point there's an error and then they fail. And now you're actually confusing the maternal with the paternal, and the two are getting jumbled up. So nearby they do well, at long distances they don't. But with local ancestry, we can correct those errors because we see them as, and I'll show you that here, we see them as simultaneous ancestry switches at the same exact position in the two different parental chromosomes. And that doesn't happen by chance, right? You don't by chance have your mother switching from, let's say, blue is European to red being East Asian, let's say, ancestry, at exactly the same position in the genome that your father happened to switch in the exact opposite direction from East Asian to blue European, right? That would not happen by chance. That's too much of a coincidence. That happened because actually this segment that's red up here belongs down here, right? It's really just your mother's segment that has been accidentally switched and put on the wrong chromosome. And so that's easy to see by us with our eyes. So obviously it should be easy for a machine learning algorithm to find those errors. And indeed they can. And this is what you're seeing here. You're seeing what we call genofix, which is Gnomix's phase error fixing algorithm playing out. And so you see initially all of these little segments that are errors, they're switched from one parent to the other. And we used a Beagle phasing algorithm here to do this, so it's quite accurate, but it still makes errors. And then you see our algorithm playing out on top of that and sorting things out properly. And at the end, you see nice, clean bars of ancestry in the two different parental chromosomes. And so this is another reason to use Gnomix, because it will run this for you. It will correct all your phasing errors, and it will give you a nice output. RFMix version 1, the first version, did this. Most people are not using that anymore because it's very unwieldy to use. It doesn't run on VCF files, etc. And in any case, the algorithm it uses to do it does not scale well at all and is completely impractical for large numbers of samples. So you wouldn't have been able to use it anyway, but this new algorithm you can use. So another nice reason to use Gnomix. And here we look at the accuracy of phasing as you go further, two loci that are further apart from each other along the genome. As you go farther and farther apart, you see that a genome that's been phased with Beagle, you've rapidly converged to just 0.5 accuracy, which is a coin flip, whether you're getting the phase correctly, whether you're on the maternal or the paternal chromosome. But with GenoFix, which is our rephasing algorithm for genomics, and with RFMix, which version 1, as I said, which is not practical to use on large numbers of samples in any case, but you get much higher accuracy of correct phasing. So it works quite well. Even at long distances. And that's what you could see visually here as the algorithm sorts things out. Okay, and finally, the confusion matrix I was promising. So here are different ancestries on the y-axis, and again, different ancestries on the x-axis, and you can see how they're being confused between each other when you run genomics. In general, you can see our confusion is extremely low. So that you see this dark color means less than 5% confusion. We generally get all the ancestries completely correct. The only ones, African here in light color of African classified correctly as African, right, and incorrectly classified as no other ancestry. These are all dark, which is like less than 1% error. The only place you see a slight confusion, as I mentioned before, is between West Asian, which is Middle Eastern, and European. And as I mentioned, it's a very subtle distinction to make between European, especially Mediterranean Europeans, and Middle Eastern. So we do very well. And even that confusion, as you can see, is not very, very strong. It's less than 20%. Okay. So then that's local ancestry, and this algorithm, as I mentioned, Gnomix, is available on my GitHub, AI-sandbox. You can run it right out of the box. It has all the default set. It works really well. You can also run it on trained models that already exist, so you don't even have to worry about trying to figure out how to get reference samples and which ancestries are which samples are completely single ancestry or anything like that. We already have that there for you. You can just download the trade model. Now, the last thing I want to talk about in the remaining 6 minutes before we go to questions is our new ancestry clustering algorithm. So if you remember, I talked about ancestry clustering as being another important technique. Ancestry clustering is where you look at the entire genome for an individual and try to look at, for that individual, what proportions do they have of different ancestries, instead of classifying each segment of their genome, which is what you see here, each segment, and classifying which ancestry it came from. That's local ancestry. Instead, global ancestry clustering, we said, was where we look at every person's entire genome as a single point, their whole genome, not looking at segments of it. And then we, for each point, for each individual genome, we say, how close is it to different cluster centers? And those are the dark bold dots here. And maybe it's halfway in between two cluster centers. And so we say that individual has half ancestry from those two sources. And it's, as I said, just really a descriptive mechanism, and gets usually represented in bar plots like this, where every individual is a vertical bar colored by the amount of ancestry they have from that cluster center, from that colored cluster center. Okay, so again, the existing methods, admixture and structure, are not feasible to run on large numbers of samples, hundreds of thousands of samples in biobanks. And so we have a new technique which uses neural networks to do this. This is called neural admixture. One of the nice things that I'm showing it right here about the architecture of neural admixture is that we can share, we basically have a single representation for the clusters, which allows us to simultaneously run many different cluster numbers. So when running admixture, and I sort of, didn't go into this in depth when I was showing you the plots, but you clearly need to select how many of those cluster centers you're going to have. If you have more cluster centers, then you're going to have more colors that you're using to color in every individual, right? And that's something you generally choose, and it's usually chosen by running the analysis with many different cluster center numbers. So you try it with 3 clusters, and you see how that looks, and then you try running with there being 4 cluster centers, right? So maybe you think that there's basically 4 population clusters going on fundamentally in your data set. You have people who came from Spain, and you have people who were already there as indigenous Americans, and maybe you have Africans who were brought as slaves, and that's your hypothesis, and so you think 3 is a good number, but you try 4 just to see if there's something else, and as I showed you in our recent paper, there is something else in Mexico. There's an East Asian cluster also from the Manila galleons that were bringing basically slaves from the Philippines, and you can see that in Pacific Mexico. So you try many different cluster centers, and you see what the representation looks like and choose from there. And the nice thing about neural admixture is that you can run it with many different cluster numbers, 3, 4, 5, all simultaneously, because the representation is for those clusters is shared. And so essentially the time that it would take to run any of the existing admixture algorithms for 10 clusters would take 10 times as long as running neural admixture, because you run neural admixture once and you get all of those. You get 2 clusters. You can also get what it would look like with 3 clusters, 4, 5, 6, 7, everything all at once. And maybe that seems like it's actually really easy, and you might wonder why people didn't do that before. Let me just make it clear, though, that when you run with a larger number of clusters, all the cluster centers shift, because the model is trying to fit the data as well as possible with that number of clusters. So the best fit for 4 is going to be different than for 3. All the cluster centers will shift. So it's not as simple as just taking the cluster centers from the previous analysis and reusing them. It's more complex than that, but we can do it, and that's part of the, this is one major speed up. But even without this speed up, we are much faster than the regular admixture algorithm, and that's here. And I'm just showing the speed up here. You focus just on the very last column. So the last column shows the speed up relative to the classical admixture algorithm. So that the speed up relative to classical admixture algorithm for admixture itself is always 1. Obviously, the classical algorithm is the baseline. And then for our neural admixture, you see that we get 114 times speed up on chromosome 1. On our simulated chromosome 22, we got 137 times speed up. et cetera. So we're talking about 2 orders of magnitude here, which makes a huge difference, right? If we're talking about something running on the data set in 1 day instead of in 100 days, taking half of the year. So this algorithm now makes it possible to run these analyses on huge biobanks. And here I'm just showing the colored clustering that you get if you use on top the old classical admixture, and on the bottom our new neural admixture, and you see it works very well. And here I plotted the cluster centers found by the classical versus ours, and ours actually performs better in some cases. It gets a more central. You see here for this cluster center, for example, ours is the square, and you see we find this cluster center right in the middle of this cluster, and you see the traditional algorithm, the triangle, found the cluster center up here, which is sort of in between the clusters. So it didn't do very well for that one. So we do as well. We're finding centers that are almost the same as the classical algorithm in most cases, and where we deviate, we do better, and we're much faster, right? So that's the overview of neural admixture, which is also available on the GitHub I mentioned, AI-sandbox. These are all methods that are out there for running, again, on large whole genome biobanks, but they work just as well with small data sets that are array data. They're more accurate even in that case. It's just that you don't have to use them. You could use the old methods, but why should you use these ones? That's what I'd like to sell this as anyway. And we tried to make these easier to use. Also, we have these saved models for the genomics. And for neural admixture, we've tried to make it really simple, and we're actively maintaining them. If you have any problems, let us know. If there's anything you think that will make it easier to use, let us know, and we can implement those changes. Because we want people to use this, and we want them to be, the simple way to go forward. So the last thing I wanted to do is thank the other authors on these. So this I didn't mention. Albert Dominguez is an amazing student who implemented this neural admixture model. He's got a lot of background in deep learning, and he did this as part of his master's thesis. And this was a collaboration we had with Barcelona, and it was a great collaboration, and I really enjoyed it. And he learned a lot of genetics along the way. I also want to thank Daniel Montserrat, who is a postdoc who worked with me on this. I want to thank Carlos Bustamante, who helped support some of this work. And then I want to thank the GSP project, the Genome Sequencing Program, which also provided me with some of the funding while I was doing my postdoc and beginning this work. And that's something that I see Emer is on the call, and she's the lead for that, for our analysis center, and I want to thank her for that. And yeah, I'll take any questions now.
Awesome, Alex. Thank you so much for that talk. This was very comprehensive, and I think you did a great job of sort of speaking at both levels for folks who are, on this call to hear about the nitty-gritty details of the methods, as well as folks who are kind of curious about kind of high-level questions. So we did have one in the chat, one of each kind of questions in the chat. I think maybe we'll start with the broader one. So from Simon. He says, wonderful talk. Thank you. For someone who's not familiar with this type of work, can you briefly explain what accuracy means? How is it judged in lay terms? I hope this makes sense.
Yeah.
Yeah.
Thank you, Alice. Yeah, I'm sorry. I always have trouble monitoring the chat at the same time as I'm talking. So I'm sorry I left.
No worries.
All right. That's a great question. So it's exactly, so what is accuracy? It's a good point, especially because, as I mentioned, the way these historical populations were formed is not one that really you can't really draw hard boundaries, and that's not the way, of course, these things work. So what we do is we take known genomes could be 1,000 genomes, could be Human Genome Diversity Project, something like that, that had an ancestral location in a certain place, and we say, okay, we're going to take that genome. So we're going to take, let's say, the Spanish genomes from 1,000 genomes as the definition of Spanish. And it may be that some of those individuals actually had some other ancestry in their genome, and so maybe there isn't going to never be possible to get 100% accuracy. But we take them, and then maybe we take some of the samples from Beijing in 1,000 genomes, and we simulate admixed progeny, and we can do that just using the combination map. And so we create grandchildren and great-grandchildren, et cetera. And now we know for every segment, because we simulated the admixture between those real genomes, we know which segments came from which of the great-grandparents, et cetera. And so we know the truth, at least in terms of which great-grandparent contributed that segment. And so then when we run the algorithm, we know whether we're finding the right boundaries for those segments. And that's how we define accuracy.
Thank you very much. And sorry for the awkward kind of question, but it's a great talk. Thank you very much indeed.
Thank you. Appreciate it. Great.
And then this technical question, Emer, looks like you're on, so maybe.
Well, I'd actually say, because I get a lot of chance to talk with Alex, so if anybody, I would totally cede my time because it's a technical question. If anybody else from this group would like to ask Alex a question.
It's fine. I think we have a few more minutes anyway, so I'll
okay,
take
well,
yep.
Well, if not, if you forgive my sort of deep delve. So one of the very interesting things that I saw in your talk, Alex, was the accuracy of detection of local ancestry tracks is very high, even in what we would normally consider very ancient timescales for detecting admixture events. So I wondered, could you, and you were going back 100 generations, and I don't know what your generation time is. I assume it's 25 or 30 years. So that's really 2.5 to 3,000 years ago. And I wanted to ask you to comment on the sort of sensitivity of your method, because that becomes very important when track lengths become very small and broken down over time. And also maybe to think about what type of admixture you could now be enabled to uncover that maybe previous methods weren't sensitive to do.
Yeah, thank you. That's a great question. I'd love to talk about that. So thanks for setting me up with that one. So with genomics, if you have whole genome sequence data, so you have a lot of very high resolution markers along the genome, so you can find very small segments accurately. We can get a 90% accuracy back to 100 generations, which is really powerful, as you're pointing out, Emer. So that means that we can look at events that happened back into the, in Europe, it would be the Roman period, even before that. So 100 generations ago is like, between 2,500 and 3,000 years ago. So we're talking about, very deep events. So what are some of those things you can look at? Well, one of the things that's interesting is, and this is relevant for Hispanic populations, Spanish ancestry itself, and I think this gets back to the previous question, is often got small segments that come from Africa. And those are very old often because they come from the period during which Spain was ruled by the Arab caliphates and Granada and all of this sort of really interesting Andalusia period that had a lot of influence from North Africa. And those segments are in Spanish genomes, and then of course in Spanish ancestry in the New World, in Latin American populations. And those events are things that happened about 1,500 years ago, and we would definitely be able to see those. That is, at that point we have like 95% accuracy. So we could find these African segments, which might be interesting in looking at admixture mapping or other things in the Spanish European ancestry that people may have in Latin America. That's one that I can think of off the top of my head. But I'm sure there's a lot of other examples of older admixture events that could be interesting to look at with this method.
Yeah, super interesting. And could you comment how long those segments are? I'm guessing they're all under 5 centimorgan.
That's right. I think you would have mostly only segments under 5 centimorgans for that. That's right. Yeah. But again, you need the whole genome to do that. With the array data. That's not going to be possible. But this is a nice feature of genomics. We really harness the power of whole genome data. So some of these older methods, they're sort of struggling with it computationally, and they're not really enabling the additional accuracy which you get from having all those extra variants, this high resolution sequence data. And that's what we wanted to take advantage of. So
great. And I think Sharon in the chats had a point to follow up on that.
Oh, great.
Yeah. And I apologize because I had a conflict, and so I missed all of it. But just the comment you just made just really strikes me that in terms of trying to explain this to individuals, this idea that, well, there's African ancestry that's recent, right? Recent in our mind maybe being slavery, right, sadly. And then there's African ancestry that actually goes back to 1,500 years ago, that I think these types of, and this has come up in Latin America with Ashkenazi alleles that go back to Columbus time. I think it sort of highlights the difficulty we're going to have in sort of explaining this type of data to individuals. Not that you're trying to do that, but I think it sort of highlights how what scientifically can be fascinating may be very difficult to explain.
Yeah, I think that's a really good point. I think one of the things that I see is combining this with other analyses, so one doesn't need to dwell really on the percents that somebody has of African or Native American ancestry, something like that, and try to instead dive straight, use these techniques to then dive straight into whatever it is that you want to do. If that's admixture mapping, like we did here at Stanford with the COVID patients' genomes, then you can do this analysis and get really interesting results of which loci are associated with something without having to really do the whole historical background and also talk to people about whether they're 0.5% this ancestry or that ancestry, and which may be more complicated to explain, as you're pointing out, Sharon, because people may wonder why am I, why are you saying that I have some African ancestry if I'm Spanish or something like that?
I'm curious about, how you think about this, particularly when we're talking about something like COVID severity and linking that to ancestry. You mentioned that it would be nice to have other kinds of data. I think it'd be really interesting for the group to hear, and I know firsthand, so this is sort of like a personal motivation that this come up, but I mean, how difficult is it to get the other kinds of data that we might want to explain some of the confounding factors that might be giving us this signal associated with ancestry, and what are some of the pitfalls, that you see with using ancestry, associations to look at traits that may or may not have anything to do with actual genetic ancestry?
Yeah, no, it's a great question, and COVID is a good place to talk about it because, of course, the main effects with COVID infection rates anyway, were who could quarantine and who couldn't, because they, were in jobs that weren't, quarantining, weren't allowing it, essential workers and others. So I think that's a great question, Alice. It's always hard to get, all the data you would like, as for, all these extra confounding factors, which of course there are many. One thing I like about the admixture mapping approach is that you could have two siblings in the same household, in the same location, going through the sort of same profession, who would have different segments of their genome coming from their parents' admixed ancestry, because it's of course a random process which segment you get from which of your ancestors. And so even two siblings will have a different shuffling along their genome of where they have an African segment and where they have a European and where they have a Native American. And so if we're looking at associations along the genome, at that level of where having a particular ancestry on your genome is associated with, let's say, more severe COVID, I think we can just naturally, through this like natural sort of randomization process, control out a lot of those variables. Now, if we're looking at overall ancestry associations, is there some association between indigenous American ancestry and COVID? Then it's going to be very badly confounded by all sorts of other things that are correlated to that. And in fact, for this plot, just to point out, we regressed out the overall ancestry. So we regressed that out. So what you're seeing here is the residual after taking out the effect of a person's overall percent from each of these ancestries. It's also the residual after taking out age and BMI and the other big factors that are driving COVID severity. But yeah, there's always extra variables you'd like to have. I think that's an important point to always keep in mind. Thanks, Alice.
Other questions, and we are here in the ClinGen context, so I would encourage folks who have questions about clinical ancestry or ideas about that to ask Alex, either about implementation or utility potentially.
I have
Alice, I think Claudia had a question there in the chat.
Yeah, I just noticed it. Thank you. So yeah, so the question is, how does admixture mapping perform when doing it on whole genome versus imputed data, realizing that there aren't many reference genomes for many of these underrepresented populations? That's a great question. I think that when you're doing imputation with very few references from populations that you think are relevant to your data set, that you have to be very, very careful. And for that reason, actually, we do have, so I have a method that I haven't fully published yet, so it needs to go up on bioRxiv, but it's here in this plot actually, and it allows you to do some ancestry, some subtle ancestry mapping without imputation. And I think ideally, if you have really underrepresented populations, these here, this was looking at ancestry and sort of Oceanian populations, which really there are not good references for. Often people, as this is an aside, but I'll just say, people think of Oceania as this one region, and it's not. It's extremely complex. You have Papua New Guinea and the Solomon Islands that Emer knows well, and all of these places which are Melanesian ancestry, and then you have different ancestry in Polynesia, very different ancestry. You have some of that, but a lot of the ancestry is coming more from Austronesian, which is completely different. And so there are just not good references from across that region right now. And so I would not really trust imputation for populations from that. And so I think the analyses need to be done without imputation. And there are things we can do, and this is a method I'm mentioning here in this paper that I developed. But I think we need to wait, in general, the answer is we need to wait until we get better references. And I would point out one other subtle detail, which is from this paper, I just want to call it out, that we just published about a month ago. And this paper focused on Polynesia. And even within Polynesia, where you might think, okay, this is Pacific Islanders, and we just need some references from Pacific Islanders, because the entire Polynesian settlement process was a series of founder events, one after the other. Small group of individuals sail across several thousand kilometers of open ocean and found a new island, and then exponentially grow in population and define a new, basically genetic pool, right? This is a huge amount of genetic drift when you have a small number of founders, right? And each island was that way. And it's a series of these. It's a series of basically telescoping bottlenecks. And we describe that very clearly in this paper. And that's what this plot on the right shows, this sort of decline of bottlenecking. Anyway, each of these island populations, therefore, is unique. And really, I think imputing the genomes of one island from genomes from even a different island 1,000 kilometers away is not going to work very well, because there were really strong founder events in founding these islands. And so you just have a lot of different frequency distributions of alleles on the different islands. So you really need good references for each of those populations. It's not enough just to call someone a single category. And again, that gets back to the danger of seeing ancestry as really a simple categorical thing. It's not.
Fantastic. Well, we are at the hour. Thank you so much, Alex, for your time and your fantastic, meticulous work on this. I look forward to seeing what comes out from you next, and see how this gets implemented. Hopefully people will use it, and I know that you'll be working hard to help people do that.
Thank you for having me on, Alice.
Of course. Have a good week, everyone.